next, we'll be hearing about the new database features. So really exciting behind the scenes look at the query API and the custom exports. And Jay is already ready for us. So I'd like to introduce Dr. Jason Greenbaum to present the query API portion. Thanks, Jay.
Thanks, Nina. So I am very excited to show you the new IEDB Query API. So it's a completely new interface to be able to query against IEDB. And so if you're not familiar with what the term API means, it stands for Application Programming Interface. And what we're doing is we're providing this alternative workflow, an alternative interface for getting at the data that's in the IEDB. So what I'm depicting here is what the current workflow is, where we have a user that goes to the web interface and they enter their query, and that web interface then talks directly to the database backend. And what we've developed now is another layer on top of that database, and it's an API layer, and it's based on the PostgreST platform. And what that does is it allows you to get at the data by writing scripts against it or using URLs to write queries. And the content of what's there is pretty much identical to the web-based version. The format's a little bit different. And the ultimate goal is to kind of remove the original interface where the web interface is going directly to the database, and instead having that also go through the API. So having kind of this common interface for both applications. So how does it work? How would you use it? If you go to this URL here, this one in green, this is kind of the base URL of the query interface, the query API interface. And when you go there, you'll be presented with a JSON file that kind of describes all of the different endpoints and how to query the different tables. So each database table that's available for querying is exposed as a separate endpoint. So you would just kind of tack on the table of interest onto your URL to query that table. So for instance, if you want to query for epitopes, you would go to the epitope search table, T-cell assays, T-cell search, and for antigens, antigen search, and so on and so forth. Likely, you don't want to query, you don't want to pull the entirety of the data down, which is what you would do if you went directly to one of these URLs, but instead you would want to filter your data somehow. And for that, you would use PostgreST's expressive syntax with something like this. So say you want to search for a particular linear sequence, you just add the field that you want to query and the value that you want to query for. And then I'll get into this kind of weird EQ operator in a couple slides. When you put that all together, you have your base URL, the table that you want to hit, and your particular query. And that should return something that looks like this. So you will get a JSON response back. And since we queried the epitope table, we're going to get back kind of one row or one record per epitope. So you can see here, there were 3 records returned from this query. And the fields that are returned are consistent between these records. The actual data that's in the fields is not the same, but the structure of those records is consistent. If we perform the same query, but now on the assay table, we're going to get an assay-centric response. So instead of 1 record per epitope, now we're going to get a response that includes 1 record per assay. And so, like Alex showed in his presentation, a given epitope can be tested in multiple assays, and so that's why we see that kind of one-to-many relationship here. And so, so what I showed you on these previous couple slides isn't the entire truth. If you just query the epitope search table, even if you add a filter on top of that, you're actually going to get back all of the fields that are in the epitope table. And so you're going to get back something that looks like this. And you probably don't, maybe you want that, but you probably don't. And you instead will want to kind of horizontally filter, or kind of shrink down the number of columns that you want returned. And you do that by using the select parameter. And what that looks like is it's just another parameter that you add onto the end of your query, and you say select equals, and then you list out the different fields that you actually want returned. And then you get back a much more manageable response. And this is what I actually executed on the previous slide. Aside from horizontal filtering, which shrinks down this way, we can also do vertical filtering, which shrinks this way. So it'll shrink your, it'll do a row-wise shrinking. You query the specific field values, and you can limit the number of records returned. So as opposed to the number of fields returned. And of course, you can combine the two concepts. And so here's an example. If we just search the epitope table, we're going to get over 1 million records returned. Again, that's probably not what you want. You want to target your search somehow. And here we're doing our synfecal query for this linear sequence, and we get our 3 records of interest. And so the search operators that you've been seeing, particularly this EQ search operator, is a operator that comes from the underlying technology that we're using called PostgreST, where it provides these different ways of comparing fields. So and you can't say directly linear sequence equals synfecal. You have to tell the system that you're searching for equality. And that's what this EQ dot operator does. And you use that EQ dot operator on fields that are numerics or strings. And then we have, there are a lot of operators, but one of the other more frequently used operators, at least in our use cases, is this contains operator. And that is kind of similar to the EQ operator, but instead of checking directly against a string field, you can query a field that contains list values. So for instance, an antigen can be associated with many epitopes, and that is encapsulated in this linear sequences field as a list. And so to search for a synfecal sequence in that list, you need to use the contains operator. And that is done just like this, contains dot, and then curly braces. And you can actually list out different linear sequences within here with comma separation. And then on the right, you can see that's the full list of the PostgreST operators. So there are many of them. By just using these two and maybe also the in dot operator, you can cover maybe 80, 90% of the things that you might want to do on our system. So the other neat thing, or one other neat thing about our system and the design of the tables, is that the system has been designed such that the query parameters are really similar across all of the tables. So when you want to pull out related information from, say, the antigen table to the epitope table, you don't really have to change your query all that much. So on the epitope table, when you want to search for the linear sequence synfecal, you do what we've been doing here, add that EQ operator. And when you want to pull out the related information in the antigens table, you just change that to contains operator. And you kind of just flip between those two operators to reform your query to pull out the relevant data for any of these tables. So why would you want to use the API? I mean, it gives you direct access to these nice formats. So the computer-friendly JSON format, human-friendly TSV formats. It will allow you to incorporate your queries into code and work with the data in your language of choice. You can use the identifiers that are in the IEDB, all the external identifiers to other resources to be able to link to those resources. You can come up with scripts that will rerun queries automatically for new or changed data, run those periodically. And we hope that it can just generally accommodate queries that aren't even possible through the website at this point. And I think I mentioned earlier, but you should be able to query just about everything that's available through the web interface. And if you can't figure out how to, please let us know and we'll try to work on the use case. We really love challenge and kind of working through these issues and making sure that the API can accommodate everybody. This is a great place to get started. If you go to this link, this brings you to our interactive documentation. So we use a system called Swagger that presents this interactive documentation that shows all of the different endpoints that are available with the tables, with a description of each table, a description of each field, and will even allow you to interactively run your queries and test them. So that's a great place to start. Here, I just wanted to show you kind of working through that interactive documentation. On the left-hand side, we're running a query through the web interface. On the right-hand side, we're at the epitope search table in the interactive documentation, and we're just searching for synfecal in both of them. On the web interface, we just have to enter the sequence. On the interactive documentation, which hits our query API, you have to use that EQ operator. You hit execute, it shows you the full URL that it's going to hit, and then it actually executes that live and will show you the response right in the documentation itself. You can find libraries to do this in all of the major programming languages. And here we just have an example of running the same very simple query using just simple GET libraries, HTTP GET libraries in these various programming languages. There are also PostgreSQL specific packages available. We haven't personally tested any of them, but there seem to be many available in different languages. So if those are helpful to you, that would be great to know. And here's just a few links that you can go to learn more about the system. I already showed you the Swagger documentation. The PostgreSQL reference is amazing, very detailed, and you can learn a lot about just working with PostgreSQL in general. We put together a few use cases on GitHub as Jupyter notebooks and R Markdown files. Some of them are just kind of contrived use cases where we're kind of walking you through the system and teaching you how to use just various operators and tables and linking things together. And some are actual user-submitted use cases. And we'd love to get yours too. And then on Zendesk, we have an article that walks you a little bit more through the system, tells you about some of the idiosyncrasies of the system, and it's another good resource. And this is the team that has been working on this. And Kelly from Lighthouse, he's been particularly instrumental in getting this done, and he is now going to tell you about the new export functionality. Thanks.
Thanks, Jay. Here, I'm going to share my screen to show some of the new export features that we're trying to implement, and hopefully it's going to be coming pretty soon. Randy earlier touched on our export features, and as she said, that it's a very important part of the website that our users use all the time and also request changes to. But currently we are limited to just these two options that you see here, the export to CSV and the CSV with IRIs, which limits our users to being able to export just a subset of fields that we determine, and only those fields, as well as only in a CSV format. So what we are working on is giving the ability to customize these exports to be exactly what is wanted or consuming the data on the end of the user. So we've coming up with this new kind of design where you click on this export, custom export option, and it pulls up this dialog here, which lets you choose multiple different options in which to export your data. I'm going to first touch on the bottom part because it's probably the most important portion of it, because it is showing exactly the different fields that you can export. Randy showed in her presentation some of these. This is the section header or the field header that she showed in her examples, and these are the fields themselves. As you can see, you can go along here and choose any options you want or don't want, or if you don't want an entire section, like if you don't care about the related object information for your epitope export, you can deselect that option and choose whatever you want to export out. What we're exploring is adding this field set option here, which gives pre-canned sets of selections that we think are important, or we determine that our users are exporting often, and you can select this to just pre-select what we determine could be important. Sorry. So for example, the base export shows what we currently export in those two options I showed you earlier. You can see that it's got all the ID fields, but it does not include the IRIs currently, because that's covered in the IRI export that we have. So if you go there, you can see instead of the IDs, you now get the identifiers, the URI identifiers for these fields. On top of that, we have the ability to change the headers from that dual header format where you see the section header and the field header as two separate rows, because sometimes you don't want to have to go through two different headers, especially if you're a programmer and you're trying to consume this data. It sometimes can get kind of confusing on how to parse that or how to filter by those rows, for example, in Excel or a program like that. So you can choose a single header, which concatenates the section header with the field header, or you can go simple no headers, where it's just pure the data, which is what you'd probably select if you wanted to feed it into a program that you've written or that you utilize, and you just want the data. On top of that, we are also expanding the formats in which you can export to. Currently, like I said, we only have CSV, which is comma separated values, which is a standard programming format where everything is divided by commas, and then it's readable, but it's not extremely readable unless you open it in something like Excel. We're adding in a native Excel feature or Excel format, which is this XL or XLS or XLSX format, which greatly expands the ability to format the data so it's more easily read by the user. That's one of the problems we've had, is how to display this information to be user readable in a nice, pretty format. And with getting the ability to natively export those to Excel formats really expands that ability to us. We're also adding tab separated format, which is just like comma separated format, just using tabs instead of commas. Again, this is another format that is used mostly by programmers or developers to consume the data. Sometimes the tab format works better than commas. And then we're also experimenting with potentially a JSON format, which is similar to what the API that Jay was talking about returns, but it's going to be a JSON representation of our current exports, or sorry, of our exports that you get from the website, as opposed to straight from the database API that the PostgreSQL database will, or API will give you. I'm going to show a couple examples here to kind of show the differences on the different formats and some of the options. So let me pull up so currently, this is what this export would look like from the IEDB. If you opened up the CSV in Excel, you can see the double headers here, as well as the field headers here. So you can see that it's replicated over and over and over and over again for each of the sections, which is a little confusing to the user, but gets across that the different sections with the limited options that we have in Excel. Let me go to another one. This is what it would look like in our current Excel one that we're working on. You can see that the headers are expanded out, so they look a little nicer, and it's more obvious that this is a section, this is a section. On top of that, we are also working on adding these additional sheets down here into the Excel exports explicitly, where we have more field descriptions to kind of give some of that information that Randy talked about in her speech, to describe what each of the fields mean in a little bit more detail, so it's not as just beyond a small little short blurb. We can explain better, as well as this ability to export the options, the query parameters that you ran your query with. So you can come back, if you save this, you can see exactly what these results pertain to in the IEDB. Let me see, you can see here the option that you can choose, so you're not strictly restricted to the options that we have currently on the website. You can pick and choose your fields as you come across them. So that's one of the biggest changes, and one of the most requested features that we have, that we get from our users for the IEDB, is the ability to customize these exports to fit exactly what they want and show exactly the data that they're interested in. And for example, one of the ones that we're also working on and that we don't offer currently is the ability to export just as you see it in the tabs. So this one is, for example, the epitope one, where you just see the details ID, the description, and then the parent protein and the parent organism. We don't offer this functionality currently, but we're looking at adding it with this redesign and this improvement. And I think that is my 10 minutes.
That is perfect timing. Thank you so much, Kelly. And thank you, Jay, before Kelly as well. I'll give everyone one more minute to submit their questions. I, again, wanted to thank our presenters, but really wanted to touch on the benefits of attending this workshop. So you've just heard about the Query API, a very exciting new feature that we've released for your use. So you've seen a bit of I guess the newest thing on the block. And now Kelly has given you a behind the scenes view of something that's still in development, and that's actually been highly sought after, as Kelly said, by our users. So it's another fantastic example whereby if our users tell us they really want something, they want to be able to customize their exports, then that ultimately gets prioritized within the team, and we work on that. And we do it through a cycle of iterative design, feedback, and so on. And we, test this through our team, through other users, through an expert committee that we have as well. So I hope you really enjoyed the behind the scenes look at something that's coming in the not so distant future. But with that, I would like to invite Jay, Kelly, and Alba back to the virtual stage for our Q&A. And Alba, I can see you'll like to answer the question first.
Yeah, thank you. I'm gonna start with the first question from Raul Acosta. How HLA restriction information availability can impact real-life immunization project? That's a very nice scientific philosophical question. I'm gonna just try to dig a little more into that, and the other ones feel free to chip in on that. But basically, as we were trying to highlight, the IEDB contains the type of information that experimentalists provide. So at the same time, the user that uses the IEDB should also be aware of the fact that the HLA restriction availability might differ. So how this can impact projects. So first of all, by doing a query and realizing that there is a bias in HLA restriction, that could be a great way to start a new project. The other way to do that is acknowledging the fact that maybe the HLA restriction are unbiased, you don't have enough information. You can always supplement that with prediction. And I guess tomorrow we will have more example of how to use T-cell prediction tools to actually try to predict HLA restriction for which there are no experimental data. And then finally, and that's something that we did the last year when we are actually curious what is recognized on SARS-CoV-2, and we didn't know anything about it. So the first way we start was to perform the IEDB query like larger. So we were looking at SARS-CoV-2, we look at the entire coronaviruses, looking for a region that maybe or restriction that were available for similar viruses to try to drag some conclusion. And I hope that might have answered a little bit the question.
And I could take the next question about the API. So I'm going to share my screen also to show an example. And so the question is, the API looks awesome. I mean, that's not the question, but thank you. What would be the best way to link to particular epitopes? If you've got a website with our epitopes, and I want to check if they're also in IEDB and provide a link, so you can go straight to the IEDB epitope summary page. Would it be the IRI or an ID? And so, yes, you can use an IRI or an ID. If you want to just go to the epitope summary page and you already know the ID, that's kind of separate from the API at this point. You would just go here where you'd say iedb.org epitope, and then the particular epitope ID. If you want to go to that, retrieve all of the information for that epitope in the API, it's pretty straightforward. You go to queryapi.iedb.org, epitope search, and the query parameter is then the structure ID, and you use that EQ operator, and you say equals 52, and you get this as a response where it shows you all of the associated information about that epitope.
All right, I'm gonna go to the next question. Taina, first, thank you very much for your nice chat comment. I'm happy that you liked the presentation. Your question is, are there plans to expand this variant mutation check framework for other pathogens in the future? And I think Nina already told you that this is great. You provide this feedback, and definitely the IEDB team is gonna take your question into account and try to address that. There's just one thing I would like to point out, that clearly the way the SARS-CoV-2 variants have been created is a little bit particular. So of course, there are gonna be issues in curating variants for any different pathogen. It really depends on the data available, but I'm pretty sure the IEDB will try to address that. Thank you. And I guess there is another question from Marzia. So about the graphs of CD8, CD4, and B-cell responses across proteins that I show, was the graph generated from all available experimental data? What if in some, the experiments were not covered the entire protein, that the immune response data were not generated from the entire protein, but only focused on some part of it? Will it affect how the graph looks like? Thank you, Marta. This is an excellent question, and let's break this in two parts. So the graph that I show you was generated from experimental data. At least I show you this in two different instances. One was based only on our experimental data, and there we use the immunobrowser standalone tool. For the second case, yes, that was based on the entire immunobrowser and experimental data that were available June 2021. In the question of what is some experiment are not covering the entire protein, I would like to go back for a second to the output of the immunobrowser and kind of like show you over here. And maybe I didn't clarify that too much, but you do actually have the information in the epitope assay counts about negative and positive. So see, this is nicely shown over here. So for instance, in the context of the spike, you can really appreciate that the entire protein has been tested. And in some region, the response is negative because you didn't see any response. So that kind of will change if you have, for instance, for a different protein, you're not going to see this negative, I would say, brown reddish color all over the protein. So that will help you understand that that region has not been tested at all. And I hope that clarifies your question.
Thank you so much, Alba. I think Kelly just wanted to add on to the previous API question. So take it away, Kelly.
Okay, thank you. Yeah, I didn't want to interrupt Jason earlier when he was talking. Oh, I need to share my screen. So when you were asking about if you're able to get a link to the IEDB epitopes, what Jason said was correct. You can use the API to search for your particular epitopes using whatever criteria you want to find. And then you can take the structure ID and put it into the API or into the URL that he showed, which is idb.org/epitope/whatever ID you would like. But the other option is, and it's kind of going into what the IRIs actually stand for. The IRIs in our system in the API are in the security standard format, which has a prefix here and then an identifier. So what that stands for is the prefix stands for a shortened version of a URL or an IRI, which is a unique identifier, which looks like a URL. And then you append on the end here. If in the API, we have this table, which is called query map, which maps these prefixes to the IRI that you would use. So for example, the IEDB epitope one that we saw here goes directly to idb.org/epitope/ then you would put, like, if you found that epitope number 52 that you're interested in, you put that on the end from the from the API. So it's kind of a shorthand so that we aren't transferring all this data all the time with any of our IRIs. And this is universal for any of the IRIs you find in the API, or for that matter, on the details pages on the website. This will be the prefix to that link so you can link to them easily. I hope that helps.
Excellent. Thank you so much, Kelly, Jay, Alba, for the wonderful presentations and answering those questions. We're at perfect timing, so I'll thank you all and move on to the next section.